Okay, so we'll just draw the nucleus really small. And then we've got mitochondria. We'll make those red. And actually, let's make the mitochondria, actually, hold on a second. Let's make the nucleus huge. There's the nucleus. Now, this isn't to scale, but at least we know that the mitochondria are a little bit smaller than the nucleus. So we know that this is the nucleus. We know that's the mitochondria. We can label it. Why it necessitates that it's red here. Hold on. Mitochondria. We got cytoplasm. All right. So we've got, like I said, I'm just kind of going off the cuff, so don't mind me. So we've got glucose. We'll just make glucose orange. Glucose enters the cell. So we phagocytose that, or actually we don't phagocytose it. We usually have channels that allow glucose to enter the cell. So we'll draw.
I started calling those the gatekeepers.
Yeah, the gatekeepers. That's a good way of seeing it. And so we got glucose. We know that glucose is a six carbon molecule. And actually, what we could do to make it even more specific, draw 4, 5, 6. And then instead of this, we can draw this as 1, 2, 3, 4, 5, 6. Okay, so 6 carbon molecule. So as soon as it enters our cytoplasm, we begin breaking it down. And I'm not going to draw all of the specific steps, but it undergoes this process called glycolysis, in which you make 2 3-carbon molecules called pyruvate. Okay. Now, and we'll just go through everything. So pyruvate. So actually, what I could do is I could type in the let's make this size 12 font. Glycolysis. Glycolysis, so like glucose lysis. And actually, I should probably label this glucose. Right. So glucose enters the cell, glycolysis happens. Now we make pyruvate, and I can label pyruvate here. Here's our pyruvate molecule. And then like if our cell is making contact with oxygen, we know that our mitochondria really, they need oxygen to undergo oxidative respiration. You know, the Krebs cycle happens in the mitochondria, and in order for things to move forward in the mitochondria, you need oxygen. Without oxygen, mitochondria isn't going to do its thing. Okay, but we're just going to go briefly over what happens to pyruvate, you know, if we have oxygen present. So oxygen's present, pyruvate is going to enter the mitochondria. We get something called pyruvate oxidation, you know, which breaks this down into acetyl-CoA. Then we go through the Krebs cycle. Remember the Krebs cycle, and then the electron transport chain. And so I'm just going to label this as, you know, Krebs cycle, electron transport chain, and then out pumps about, you know, roughly 36 ATP. Hold on. Okay, so that's in the presence of oxygen. oops, whatever, oxygen. There's supposed to be an O there.
Gotcha.
Of Zygen. Okay, so that's in the presence of oxygen. You know, pyruvate goes in here, pyruvate oxidation, you get the Krebs cycle. You extract those, you know, NADH, and you have FADH2. Remember the whole process of, you know, like through glycolysis, remember we're mainly extracting these electrons in the form of NADH, right? So, and actually just to add to glycolysis, like I know we make pyruvate, but we're also using, and actually I'll write it this way. We take NAD plus, that's fed into glycolysis and out pumps NADH. And remember, NADH, that's what we're sending over to the electron transport chain, you know, in the mitochondria. So we're sending usually that NADH to the electron transport chain to drop it off in the mitochondria to create that 36 ATP in the presence of oxygen. But remember, we're using NAD+. NAD+ is being converted into NADH. Now let's say we have no oxygen. Let's say, you know, we're working out and yeah, we're working out a lot and we're using oxygen, but then we're working out so intensely that our muscle cells aren't getting enough oxygen for as much energy as needed, or as much oxygen as needed by the mitochondria to keep up with that metabolic process. So what we will resort to if we're not taking in oxygen, or let's say you're working out and you're holding your breath, you know, we still need to produce energy in our cells, in our muscle cells specifically. And so what we'll do is, we don't have any oxygen, we're going to undergo a different way of producing energy. And so remember, we got pyruvate, which is this 3-carbon molecule from glycolysis. And remember, glycolysis, and I guess I should have included this too, you know, we take, you know, NAD+ and also now it's getting kind of like crowded here, huh? ADP, hold on, maybe we'll do it from the other way. ADP.
Which is because the third phosphate has broken off.
Yeah, so
So
what we're doing is, yeah, what we're doing in glycolysis is we're taking ADP and we're making ATP, remember? So we'll just say ADP, and then out comes, that goes into glycolysis. And remember, we're netting 2 ATP. So I'll just put ATP. So I guess those are some important things that I need to mention. You know, you start off with the glucose. Glucose is broken down into pyruvate, 2 molecules of pyruvate. But importantly, we're using NAD+ to create NADH, and we're using ADP to create ATP. And so remember, this is going to make a little bit of energy. And, you know, with oxidative respiration, you're making about 36 ATP per glucose molecule. When it comes to glycolysis, you're making about 2 ATP per glucose molecule. So we'll just say around 2 here, around 2 ATP per glucose molecule. Now, it's not very much, but if we have no oxygen, it's our only option. Okay, so we take that ADP, we make 2 ATP. We have no oxygen, we're going to have to do something about this. So what we do is we take this pyruvate, so these pyruvate molecules, and we have them undergo a process called fermentation. And so fermentation is a very short process in us, especially. So fermentation. And so we take this pyruvate, we ferment it, and what happens is we will create. So ferment this into a 2-carbon molecule called lactic acid. Make that orange. There we go. So we're creating lactic acid. Now, in order to make this lactic acid, this is where it becomes kind of inefficient. Remember, we're creating this NADH to create the electron carriers to bring to the electron transport chain so that the mitochondria can use those electrons in the electron transport chain to make lots of ATP in the presence of oxygen. If we don't have oxygen, our mitochondria is going to be offline. Okay. So what we do is we grab the NADH that we make in glycolysis. And this is kind of like this intermediate energy currency. We're going to use this potential energy currency to drive forward fermentation. So we take that NADH that we produced in glycolysis and we use that in fermentation. So we oxidize it. Remember, NADH is going to be the reduced form containing the electron. We oxidize it. So we strip away its electron and we create NAD+. Now, this is important because in order for glycolysis to occur, we need NAD+. I'm sorry, there's a lot of like arrows here. I hope this isn't too
No, I'm with you
confusing
so
for
far.
You're with me so far. Cool,
yeah, I'm
cool.
with you. Yeah.
All right, cool. So remember when glucose enters the cell and we wanted to undergo glycolysis to make that 2 net ATP, we use NAD+ to drive it forward. And what we're doing here in fermentation is we're taking that NADH that we make and we're turning it back into NAD+ so that we can plug it back into glycolysis. So this NAD+ now moves back to glycolysis so we can create that 2 ATP again. So what's happening here, just to simplify this, fermentation potentiates another cycle of glycolysis by replenishing NAD+. And so the process that we're doing this through is we are oxidizing. So we oxidize the NADH. We oxidize, we could say, NADH into NAD+. That's important to know. That's going to be on the test because I'm going to have a question saying, you know, what's oxidized in fermentation? We're oxidizing NADH into NAD+.
Don't mind me just highlighting that. Make sure I remember. Gotta be on the test.
That's all I really need you guys to know for fermentation.
Okay.
It's a short process. You're moving, you know, you're converting pyruvate into lactic acid in terms of mammals. Like if we're talking about us, we're converting that into lactic acid and also oxidizing NADH into NAD+.
Okay. It sounds very simple when you say it, but I feel like when I was reading through it, I'm like, my brain hurts. But I also was probably tired. So,
yeah.
you know, I was like, okay, we're going to have to revisit. And
yeah.
So that makes a lot more sense. I feel like you put it in a condensed but straightforward way for me to wrap it and put it in here. So
good.
That helps. Thank you.
All right. And then I'm gonna look at what else we have here because I think what I do is in that video I'm gonna upload, and I could actually just send it to you specifically after this. I'm gonna post an announcement for it on Wednesday, but I could send it to you because I go over a lot of the study guide.
Okay, that would be perfect.
Yeah. Let me. Oh yeah, share this. Okay, can you see
the study guide? Yeah.
Study guide. Okay, cool.
Which I printed mine out, but don't ask me if I have it right in front of me right now. I know I have it somewhere.
You have what?
I said, which I printed it out. I always like to print it and then like check off what I've.
Yeah, check off what you know.
But my toddler has been on my desk and I don't see it right away. So I'm going to say it's somewhere.
Yeah. Yeah, so I think I actually go over chapter 6. I go over this whole part. So I write out the reaction for respiration. I define aerobic and anaerobic. Let's look at photosynthesis a little bit. So the right overall equation of photosynthesis, and they will oxidize and what's reduced.
I think this was my shortest of notes. So yeah, if you want to give me some extra, I will take it.
When chloroplasts of light reaction occur. Okay, we could hold on. Let me pull up. I'm going to pull up the video really quick, and I'm going to try to cover what I don't cover on the video. Actually, what I could do. Let me log into my YouTube. Switch account. My videos. And oh, you know what? I don't even have to log in. I can just go to the previous office hours recording. Okay, here we go. So let's see what I go over before. Yeah, so I go over all of chapter 6. What I can do for you is I can go over chapter 7. I'll go over like the.
Okay, sounds good.
And let's see if I can grab the chat. I'll just post a link to it in the chat. Okay, did you get that?
I did.
Cool.
And I have pulled it up. Let me pause it so it doesn't talk over you.
Okay. All right, so you can just see the study guide right now, right? Because yes. I have the chat over on the side. Okay.
Yes.
So define photosynthesis: chlorophyll, photon, proton. Cool. Photosynthesis. That's just going to be the conversion of CO2 gas in the atmosphere into usable glucose to sugar. We'll say into sugars. I would say that's the simple way of putting photosynthesis. Conversion of CO2 gas in the atmosphere into sugars through the use of light energy. That's kind of the driving force of it all. Using light energy. Okay. Chlorophyll. So chlorophyll, you could say chlorophyll would be the photopigment that aids in photosynthesis. Photon and proton. So these are two words that kind of sound similar. Photon is a packet of light energy. And a proton is an H+ ion. Fun fact about H+ ions and why they're named protons is, if you remember from the chemistry portion, your hydrogens will have one proton in the middle, no neutrons, and usually one electron on the outside. And so what happens if you make an ion out of it? By taking away its electron, all you're left with is literally a proton, which is its nucleus. So that's why we call H+ ions protons, because literally they're just a proton.
Gotcha.
Yeah. And usually, you know, the higher the H+ concentration, the more acidic something is. Okay, now the products and reactants for photosynthesis. So here I could just write the equation. So photosynthesis is going to be, you got sunlight.
Water and carbon dioxide.
Yes, H2O plus CO2. And that's going to make sugars plus, you know what else we make? Sunlight plus H2O plus CO2.
Oh, oxygen.
Yep, plus O2. What plants will usually produce. No, it's all right. Okay. Right. So remember, sunlight's going to be kind of that driving force. And the way we're getting our electrons for this whole process is we're stripping it away from H2O. So what we're doing is we're oxidizing H2O. I'll make H2O red because we're oxidizing it. And we're taking the carbons from CO2 and we're splicing it together to make sugars. So we're reducing CO2. And I'll make that green because we're reducing it. So we're oxidizing H2O, reducing CO2. CO2 is reduced into sugars. H2O is oxidized into O2. Okay. Be able to write the overall equation of photosynthesis and label what is oxidized, what's reduced. That's pretty much what we did here.
Which is it the 6 CO2 plus the 6 H2O?
Yeah, I forgot to put that in there. Yeah.
Oh, okay.
There you go. Yeah. So be able to write the overall equation. Yeah, so we just did that. Write the sub-steps of the light reaction and the Calvin cycle. I don't know if I have you guys go too deep into that because that's pretty complicated. So the light reaction, let's see. I'm going to try to simplify it. I'm going to try to simplify it here just into steps. So light reaction, what's going to happen here is you get light hits photosystem. And actually what you could say, you could say antenna. You could say the antenna complex. I mean, you can have light that hits the main photopigments, chlorophyll, which is going to be the reaction center. But the thing is just based off of pure statistics, your antenna complex takes up more surface area. So likely you're going to hit your antenna complex, which transfers energy to reaction center. And that is going to be your chlorophyll.
Bless you.
Bless you. Okay. So transfers energy to your reaction center, which we call chlorophyll. Okay.
Yes, gotcha.
Then from your reaction center, you emit. So your chlorophyll releases an electron. We'll just have that labeled as an lowercase e with a minus by it. Releases an electron. Then that electron moves. So I'll just be more specific. The electron moves through an electron transport chain, kind of like you'd have in your mitochondria, where you have an electron moving from protein to protein to protein. Pumping protons into the thylakoid space. If you remember in the electron transport chain, you're pumping hydrogens into the intermembrane space. But when we're talking about photosynthesis and the light reaction, what you're doing is you're pumping protons from your stroma into your thylakoid space. So now you have those little pancakes, they're very acidic on the inside. Now those electrons, or sorry, those hydrogens, the hydrogens drive forward ATP synthase to make ATP. Now we know that in photosynthesis, this isn't our main energy producing thing. Remember, we're trying to make sugars. We're not trying to make ATP here. But through the light reaction, we make some ATP because that ATP is necessary for the the dark reaction or the Calvin cycle, which we haven't even talked about yet. Yeah. Now, instead of, and we'll just put it here. Remember, in the electron transport chain within oxidative respiration in the mitochondria, your oxygen is the final electron acceptor in that electron transport chain. However, in photosynthesis, we have NADP plus is the final electron acceptor, forming NADPH. So end result, we have ATP and NADPH, which are these intermediate energy sources to be used in the Calvin cycle. So these are kind of like you're producing this little energy. You got a little bit of electrons. This is all going to be used in the Calvin cycle. So I would say that those are kind of the steps of the light reaction. So that was the light reaction here. I'll bold that. So now we go to Calvin cycle. Now, Calvin cycle, I'm not going to have you guys remember too much of the specific steps of the Calvin cycle. Kind of like I don't really want you guys to remember all the steps of the Krebs cycle within cell respiration. But there are some enzymes and stuff that I want you to know. So like, for example, in the Calvin cycle, your rate limiting enzyme is Rubisco, which is hypothesized to be one of the most abundant enzymes on Earth. That's just because we have more plants and photosynthesizers than we have things that eat them. We have to. If we had less photosynthesizers than we had things that ate them, then we would go extinct because we'd eat all of them, right? So more photosynthesizers on Earth. Photosynthesis requires Rubisco, so probably the most common enzyme on Earth. So the rate limiting enzyme is Rubisco within your Calvin cycle. And then I'm going to truncate this and I'm going to say ATP and NADPH generated in the light reaction is used, Calvin cycle, yeah, the rate limiting enzyme is Rubisco, ATP and NADPH generated in the light reaction is used to drive forward the Calvin cycle. And actually, importantly, we should say in the stroma. That's going to be on the test. Where does the Calvin cycle happen? In the stroma. Where does the light reaction occur? In the thylakoid membrane. So we'll say hits the antenna complex on the thylakoid membrane. Transfers energy to the reaction center, blah, 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 blah. Okay, so I'll just bold thylakoid and we'll make it orange because that's important. And then here, you know, Calvin cycle is going to happen in the stroma. So I'll make that orange too. So drive forward the Calvin cycle. You can have the Calvin cycle occurring at night. You can have the Calvin cycle occurring during the day. You could take that ATP and NADPH, as you're making it in the light reaction, start making sugars out of CO2 in the atmosphere during the day. Oftentimes what will happen is you'll store that CO2 during the day. If you're like a CAM plant or a C4 plant, and you'll store that CO2 from your stomata being open, and you'll store the ATP and NADPH, and then you'll synthesize the sugars at night. So ATP and NADPH generated in the light reaction used to drive forward the Calvin cycle in the stroma to make sugars from atmospheric CO2. All right. So we probably covered a lot there where these questions, we pretty much answered these questions. So next we have be able to distinguish the stroma, the grana, the thylakoid membrane of the chloroplast. So thylakoid membrane, that's going to be where the light reaction occurs. Your grana, that's going to be a stack of thylakoids. So I'll write that, grana, stack of thylakoids. And then your stroma. Oh yeah, do you have a question?
No, I'm trying to double check because I was like, I think Grana, I remember that word, and I'm like, I'm pretty sure I wrote that down somewhere. I was just looking through my notes. I'm sorry.
Yeah, no worries. So your stroma, that's just going to be the juice of the chloroplast. That's going to be kind of like your cytoplasm or your cytosol, of your cell. And that's going to be where your Calvin cycle occurs. So stroma is the juice of the chloroplast. That's where your Calvin cycle occurs. And then you're within your thylakoids, you could say within your thylakoid membrane, or on your grana, that's where your light reaction is going to occur. Which color of light do plants not absorb and thusly reflect? That's going to be green.
Green.
Yeah.
Which I thought was interesting. I did not know that in all my years of life.
Yeah.
It's like, wait, what?
Yeah, any colors you see, it's not because it is that color, it's because it's absorbing every color but that color, therefore reflecting it. Yeah.
Which I feel lied to because that means that water is not blue.
Yeah, it's just reflecting it, right?
Yeah. I was like, when I say I had to go,
yeah,
what?
Changes your perspective.
A little bit.
All right, so where in the chloroplast does a light reaction occur? That's going to be in your thylakoid membrane or in your thylakoid. Where does the Calvin cycle occur? In your stroma. What are the parts of a photosystem? We would say antenna complex plus the reaction center, which is typically going to be your chlorophyll. So antenna complex plus the reaction center. That is what your photosystem is composed of. What enzyme helps make ATP in photosynthesis? Just like in your electron transport chain within cell respiration is ATP synthase. What is the electron carrier made by the light reaction used in the Calvin cycle? NADPH. So it's like NADH, which is an electron character. I like to associate this P for photosynthesis, so NADPH. You're not going to find any pH in your mitochondria, only in your chloroplasts. What are the reactants and products of the Calvin cycle? So, the Calvin cycle. So the reactants would be CO2, reactants, CO2, ATP, NADPH. So those are things going into it. That's what we mean by reactants. What's going into it? And then the products, sugars. What are stomata? So those are going to be the little pores. Stomata, pores of the leaves. Sure, you can say pores of leaves, pores of plants. It's the little openings that you find on the leaves. And the functions, gas exchange. And ideally, you want CO2 in and O2 out, ideally. You want to take in that CO2 so you can make sugars, and you want to release that O2 from the breakdown, essentially, of water. How does the carbon cycle work? So this is different from the Calvin cycle. This is different from, this is kind of like a global thing. So the carbon cycle is, photosynthesizers produce oxygen, and this is very truncated. Photosynthesizers produce oxygen, and then you have your heterotrophs consuming, breathing in that oxygen and producing CO2, and then that CO2 is taken up by photosynthesizers. So that's the Calvin cycle. So, we could say heterotrophs produce CO2 and take in O2. And then your autotrophs, like your photosynthesizers, we'll put that in there. Photosynthesizers produce O2 and consume CO2. And in short, that's pretty much the carbon's, oh, I guess they can't register photosynthesizers as a word. So your autotrophs will produce CO2 and consume CO2. And we know that, what we like to do is we like to do things that kind of like offset this exchange. So now we're producing a lot of CO2, whereas, we still kind of have the same amount of producers. And actually we have less producers now because we're killing all of it.
Deforestation.
Yeah, deforestation and stuff like that. So, the long-term consequences, we haven't been able to see it firsthand in the sense of, how devastating it can be for our species. We haven't seen like the death of our species as a result yet. So this is kind of like an N of 1. This is our first experiment where we get to see what happens, the long-term consequences, unless we change something. And then the greenhouse effect. So this is where, CO2 is a greenhouse gas. If we produce a lot of CO2, it accumulates in the atmosphere and it prevents the escape of heat from the sun out into the atmosphere. So it traps it on Earth. And so that could lead to global warming. All right.